Genome editing refers to the precise modification of an organism's DNA using molecular tools such as CRISPR-Cas9, TALENs, or zinc finger nucleases. These technologies enable scientists to make targeted changes to specific genes, including insertion, deletion, or replacement of DNA sequences, with unprecedented precision and efficiency. Genome editing has the potential to revolutionize biomedical research, agriculture, and other fields by providing new opportunities for understanding gene function, treating genetic diseases, improving crop traits, and enhancing animal welfare. However, genome editing also raises ethical considerations and concerns, which revolve around issues such as unintended consequences. Genome editing technologies may introduce unintended genetic changes or off-target effects that could have unforeseen consequences for organisms and ecosystems. Ensuring the accuracy and specificity of genome editing techniques is critical to minimizing potential risks and uncertainties. Germline editing and inheritable changes. The ability to edit the germline cells, sperm, eggs, embryos, raises ethical questions about the heritability of genetic modifications and the potential long-term implications for future generations. Ethical concerns include the possibility of unintended genetic alterations, unpredictable effects on offspring, and the potential for unintended consequences to be passed on to future generations. Ethical use in humans. Genome editing raises ethical questions about its use in human embryos, particularly for purposes such as preventing genetic diseases or enhancing desirable traits. Ethical considerations include concerns about safety, consent, equity, social justice, and the potential for misuse or exploitation of genome editing technologies. Genetic enhancement and eugenics. The prospect of using genome editing to enhance human traits or capabilities raises ethical questions about the ethics of genetic enhancement, the pursuit of designer babies, and the potential for exacerbating existing social inequalities. Ethical considerations include concerns about fairness, equity, autonomy, and the social and psychological implications of genetic modification. Regulatory oversight and governance. Genome editing technologies raise questions about regulatory oversight, governance, and accountability to ensure responsible and ethical use. Ethical considerations include the need for transparent and robust regulatory frameworks, public engagement, stakeholder consultation, and international cooperation to address the ethical, legal, and social implications of genome editing. Dual use and biosecurity concerns. Genome editing technologies have dual use potential, meaning they can be used for both beneficial and harmful purposes. Ethical considerations include concerns about biosecurity, bioterrorism, and the potential misuse of genome editing tools for nefarious or unintended purposes. Environmental and ecological impacts. Genome editing of organisms for agricultural or environmental purposes raises ethical questions about the potential ecological and environmental impacts of genetically modified organisms, GMOs. Ethical considerations include concerns about biodiversity, ecosystem resilience, unintended consequences, and the precautionary principle. Addressing these ethical considerations requires careful deliberation, interdisciplinary collaboration, and engagement with stakeholders, policymakers, ethicists, scientists, and the public. Balancing the potential benefits of genome editing with its ethical, social, and environmental implications is essential to ensure responsible and ethical use of these transformative technologies.